You know, right now, in pregnancies all over the world, there's this microscopic, highly orchestrated, and honestly surprisingly violent invasion taking place.
Yeah, violent is definitely the right word for it.
Right, because we tend to think of pregnancy as this very passive, nurturing stage, just this peaceful incubator. But if you look at the cellular level, fetal cells are actively marching out of the placenta. They're invading the mother's uterine tissue, ripping up her blood vessels, and completely rebuilding them.
It's incredible. And if either side makes even a single mistake, if the invasion goes too far or if the mother's defenses fight back a little too hard, the consequences are just immediate and life-threatening.
Exactly.
It completely shatters that illusion of biology as some peaceful, static blueprint. We are looking at a landscape that is fiercely competitive. It's fluid, and it's constantly moving. You have two entirely different genetic instructions trying to build a single functioning system without rejecting each other. It is the absolute definition of biological muddy waters.
Which brings us to the focus of today's deep dive. We're looking at a frankly groundbreaking study published in the journal Nature in April 2026. A massive team of researchers set out to build this comprehensive single-cell spatial atlas of what they call the human maternal-fetal interface. And they didn't just take a snapshot.
Right, no, they tracked it over time.
Yeah, they tracked this interface from early pregnancy around gestational week 5 all the way to term at week 39.
They essentially created a submicrometer map of a temporary organ. I mean, the placenta and the maternal tissue it invades form this entirely transient, disposable life support system. And this study maps exactly how that system is built, literally cell by single cell.
Okay, let's unpack this. Our mission today is to explore exactly how maternal and fetal cells negotiate this incredibly high-stakes biological real estate. We're going to look at how cells choose their identities, how they physically alter the mother's body, and most importantly, what this new cellular map reveals about the hidden roots of severe pregnancy complications. Things like pre-eclampsia and preterm birth.
Which is huge.
It is. So whether you are, casually curious about how human life begins or you are actively studying developmental science, this deep dive is going to fundamentally change how you view the boundary between a mother and child.
Absolutely. But before we can understand the intricacies of that negotiation, we really have to grasp the sheer complexity of the terrain. Biologists refer to the maternal-fetal interface as a transient hemiallogeneic amalgam.
All right, let's translate that for a second. Transient meaning temporary.
Right.
Hemi meaning half, allo meaning other, and geneic meaning genetics. So it's a temporary merging of two half-different genetic identities, the mother and the fetus.
Exactly.
It sounds like two different countries sharing a border wall, but they are both actively dismantling and rebuilding that wall at the exact same time.
That is a highly accurate way to visualize it, and mapping that border wall has historically been almost impossible.
Because it's just a mess of cells.
Yeah, the cells are completely tangled up. So to solve this, the researchers threw just a staggering amount of technology at the problem. They looked at the RNA using single-nucleus RNA sequencing, which gives us the transcriptomes.
The active genetic instructions.
Exactly, what the cell is actively doing.
Yeah.
But they combined that with ATAC-seq. That's a test that measures chromatin accessibility.
Chromatin accessibility. So, if a cell's DNA is like a massive library of instructions, chromatin accessibility tells us which specific books are currently open on the table and ready to be read.
Yes, perfect analogy. It tells us what the cell is preparing to do, not just what it's currently doing. And they did this on over 191,000 individual nuclei.
Wow, 191,000.
Yeah. But the real breakthrough was combining all that genetic data with advanced spatial mapping. They used stomic stereosec to map over 1.1 million cells at a 0.5 micrometer resolution.
Okay, I want to put that into perspective for you. A typical human cell is maybe 10 to 30 micrometers across. So a 0.5 micrometer resolution is capturing details smaller than the nucleus of a single cell.
It's incredibly precise.
It's like upgrading from a regional paper map where you can only see the major highways to Google Street View. We aren't just getting a directory of who lives in the city. We can see exactly which cell is parked in which driveway and who their next door neighbor is.
What's fascinating here is how they figured out who belonged to which country in that border wall analogy. Because when you take a microscopic slice of this tissue, maternal and fetal cells look incredibly similar. And they're completely mixed together.
So how do you tell them apart?
The researchers used a computational algorithm to sort them out, affectionately named supercell.
Supercell, like a genetic soup.
Yeah, exactly. It essentially acts like a microscopic detective. It clusters cells by their genotype without even needing a reference genome. It basically scans for tiny, single-letter typos in the DNA code between the mother and the fetus.
That's brilliant.
And to prove this actually worked, they tested it on pregnancies with male fetuses. They just looked for the Y chromosome. If the cell had a Y chromosome, it was 100% fetal. Using this method, they managed to definitively tag over 95% of the million-plus cells as either maternal or fetal.
I can imagine the resulting map must have been pretty chaotic. Like, when they separated them out, did the cells segregate themselves? Mother cells on one side, fetal cells on the other?
Not at all. It depended entirely on the cell type. Some cells, like the fibroblasts, the connective tissue cells that provide structural support, they were a complete mix. You had maternal and fetal versions freely mingling in the exact same areas. But the vascular endothelial cells, the ones that line the blood vessels, they formed entirely distinct separate maternal and fetal clusters. They just refused to mix their molecular identities even when they were sitting right next to each other.
Which makes sense. Considering the sheer mechanical stress those blood vessels are under, you wouldn't want a mismatched pipe, right? Now, to really understand this dynamic, we need to introduce the fetal champions of this story, the progenitor cells.
Right, the stem cells. We call them villous trophoblast, or VCTs.
VCTs, got it.
They're a specific type of fetal stem cell, and they face a critical, irreversible choice early in the pregnancy. They essentially stand at this biological crossroads and have to take one of two paths.
Path A, they become the peacekeepers.
Exactly. We call those syncytia trophoblasts, or SCTs. These cells actually fuse together to form a massive multinucleated barrier. They handle the peaceful trade across the border. They absorb nutrients, process waste, and secrete the hormones necessary to maintain the pregnancy.
Just keeping the peace, paying the bills.
Yeah.
But then there's path B, the invaders.
The extravillous trophoblasts, EVTs, their entire biological purpose is to march out of the safety of the placenta, physically invade the mother's surrounding tissue, and hunt down her blood vessels to remodel them.
So if I'm tracking with you, we've got this incredibly high-resolution map, and it's showing us that these fetal stem cells, these VCTs, essentially have to pick a lane, peacekeeper or invader, and there is zero room for error.
None at all.
Which raises a massive question. How does a single cell actually make that irreversible decision? I mean, how does it know to become an invader without getting confused and trying to be a peacekeeper halfway through?
This is where the study dives deep into the epigenome, that library of open-closed DNA we talked about. By mapping the open chromatin to specific enhancers, the researchers discovered what they call a bistable toggle switch controlling the cell's fate.
A toggle switch.
Yeah. They found a specific group of biological formin, known as transcription factors, that show up and start flipping that switch.
I want to pause on that term, transcription factors. These are the proteins that bind to the DNA and basically yell the orders, right? They tell the cell, turn on the genes to build legs so we can move, or turn on the genes to make hormones.
That's exactly their job. And in the cells that decide to become the invading force, the EVTs, a very specific crew of these transcription factors, gets highly upregulated. We're talking about factors like FOS, ASCL2, KLF6, and STAT1.
Okay, so this specific crew of formin takes over.
Right. And they immediately turn on the genes required for an invasion. For example, they activate a gene called FN1, which allows the cell to move and physically grip the surrounding tissue.
I imagine they also have to turn on some kind of defense mechanism, though. Because if a fetal cell is just marching right into the mother's tissue, why doesn't her immune system immediately recognize it as foreign DNA and just destroy it?
That is one of the great mysteries of pregnancy, and this toggle switch plays a huge role here. The transcription factors turn on specific genes that produce a protein called HLA-G.
HLA-G.
Yes. This protein sits on the outside of the invading fetal cell and essentially acts as a chemical invisibility cloak. It sends a signal to the mother's patrolling immune cells that says, "Don't attack me, I belong here."
Wow. So the formin turn on the movement engines and the invisibility cloaks. But you called it a bistable toggle switch. What's the other half of the toggle?
This is the genius of the system. It's a dual action. These transcription factors don't just turn on the invader genes. They actively hunt down the genes that belong to the peacekeeper pathway, and they suppress them.
Wait, really?
Yeah, they associate negatively with the alternative fate. They actively suppress SCT genes like CGA and TFPI2.
So it's not just like turning on a light switch in a room. It's a mechanism where turning on the kitchen light automatically cuts the power to the bedroom.
Precisely.
By activating the invader sequence, they are literally suffocating the peacekeeper sequence.
It enforces total, brutal commitment. A cell cannot survive if it gets stuck halfway between those two identities. It has to lock the door behind it. Now, the study did find one really fascinating exception, a single transcription factor called GCM1.
GCM1, what does that do?
It's actually required for both pathways.
Oh, like a double agent.
You could view it that way. If a cell loses GCM1, it completely fails to become either a peacekeeper or an invader. But what the researchers found is that this single protein targets completely different gene sets depending on which lineage the cell chose. It adapts its function entirely based on the chosen fate.
That level of regulatory rewiring is mind-blowing.
Okay, so the switch is flipped, the kitchen light is on, the bedroom power is cut. Our little fetal cell has committed to becoming an EVT, an invader. It has its movement engines running, its HLA-G invisibility cloak on. It has to physically leave the placenta and crawl into the mother's tissue.
Yeah.
If we look at our Google Street View map, what does this invasion actually look like in space?
Well, the researchers mapped out distinct spatial communities in the mother's tissue, which is known as the decidua. They found that the superficial decidua, the layer closest to the placenta, is absolutely teeming with these invading EVTs. They are everywhere.
It's just a massive front line of troops. Exactly. But as you move deeper into the mother's tissue, the landscape shifts dramatically. The deep decidua is heavily dominated by the mother's immune cells and stromal cells.
Which makes sense. The deeper you go into maternal territory, the more of her security forces you encounter. But the invading fetal cells aren't just wandering around aimlessly, right? They have a highly specific target.
They do. They are hunting for the maternal uterine spiral arteries. This leads to one of the most critical phases of human development, arterial remodeling. The EVTs have to find these maternal arteries, invade them, and completely reconstruct them into low-resistance, high blood flow vessels. If this process fails, the fetus simply will not get enough blood to survive.
They essentially have to tear up a narrow, winding country road and pave a massive, high-speed highway.
That's a great way to put it.
But to do that, they have to deal with the mother's endothelial cells, the AECs, right? The cells that currently line and protect that road. Hold on, do the fetal cells kill the mother cells, or do they just shove them out of the way?
That has been a long-standing debate, and this spatial atlas provides a stunning answer. The maternal cells aren't just killed off. The researchers watched them transition through a series of transient vulnerable states.
State changes, like what?
They categorize them as R0, R1, and R2 states. It's essentially a step-by-step molecular breakdown of the maternal cells losing their grip on the vessel wall. During this remodeling, the fetal EVTs literally displace the maternal AECs. They shove them out of the way, take their jobs, and line the blood vessels themselves. You end up with a scenario where fetal cells are physically forming the walls of the mother's own blood vessels.
Here's where it gets really interesting, though. We have these fetal cells that are armed with invisibility cloaks, aggressively marching into maternal territory, ripping up blood vessels, and shoving the mother's cells out of the way through these R0, R1, R2 states. If they are that aggressive, what stops them? Why don't they just keep invading right through the wall of the uterus?
Because the mother's body is not defenseless. The study identified a highly specific subtype of maternal decidual stromal cell positioned to the deeper tissue. They call it DSC3.
DSC3, the peacekeepers on the maternal side.
Precisely. They act as the mother's brakes. And the mechanism they use to stop the invasion is entirely unexpected. These DSC3 cells actively suppress the fetal invaders using endocannabinoid signaling.
Wait, endocannabinoids, as in the exact same chemical system in the brain that responds to cannabis.
The very same system. We normally associate endocannabinoids with the nervous system, regulating pain, mood, or relaxation. But here, the mother's body is producing its own natural cannabinoids, and these specific DSC3 braking cells are using them as a chemical red light.
That is wild.
They release these signals, which bind to receptors on the invading fetal cells, essentially freezing their movement machinery. It tells them, you have gone exactly far enough, stop.
That is an incredibly elegant check and balance. A chemical stop sign deployed at the exact right micrometer.
It's a delicate, violent, and perfectly timed dance. The fetal cells must be aggressive enough to secure a blood supply, but the mother's cells must be strong enough to halt the advance before it causes massive internal damage.
Which forces us to ask the uncomfortable question. What happens when this perfectly timed negotiation fails? What if the mother's brakes are applied too early? Or what if the fetal invasion is too weak? If this biological border wall isn't built correctly, what are the real-world consequences for the pregnancy?
That leads to what is arguably the most clinically vital part of this entire study. The researchers wanted to map disease. To do this, they took their high-resolution cell-by-cell map and they integrated it with massive genetic databases known as genome-wide association studies, or GWS.
So a GWS is where scientists look at the DNA of tens of thousands of patients who all suffer from a specific disease, and they look for common genetic mutations or flaws among them, right?
Exactly. Imagine taking a map of a city and overlaying a map of every car crash that has happened over a decade. You would very quickly see which specific intersections are inherently dangerous. The researchers took genetic risk data from tens of thousands of women who suffered from major pregnancy complications, and they overlaid it onto their single-cell map. They generated trait-relevant scores to see exactly which specific cells at the interface were harboring the genetic flaws. Okay, let's talk about pre-eclampsia, because the stakes there are incredibly high. For those who don't know, it's a severe condition characterized by dangerous high blood pressure, protein in the urine, and it can lead to organ failure for the mother or fatal outcomes for the baby. For decades, the medical field has debated, is pre-eclampsia a problem with the baby's placenta, or is it a problem with the mother's vascular system? And this atlas provides a stunning, definitive answer. It's both, and in highly specific ways. When they overlaid the genetic risk for pre-eclampsia, they found that on the fetal side, the flaws were heavily concentrated in a specific subtype of the invading cells called IEDTs, the ones responsible for tearing up the roads.
So the fetal construction crew has faulty genetics.
Yes, but on the maternal side, the genetic risk was highly concentrated in three specific places. The maternal arterial endothelial cells, the ones being displaced in the blood vessels, the maternal T cells, which are immune cells, and crucially, the maternal DSC3 cells.
The ones using the endocannabinoids, the breaks themselves are faulty.
Exactly. It is a catastrophic breakdown in communication. It is not just a fetal problem or a maternal problem. The invaders are flawed and the breaks controlling them are flawed. The negotiation fails, the blood vessels aren't remodeled correctly, and the mother's blood pressure skyrockets as a result.
That fundamentally changes how we would even begin to approach a treatment for preeclampsia. But what about other complications? Did they map the genetics for things like spontaneous preterm birth, where a baby is born weeks or months too early, or sporadic miscarriages?
They did, and this is where the data revealed a massive paradigm shift. When they mapped the trait-relevant scores for both preterm birth and miscarriage, they didn't find the primary genetic flaws in the mature placenta or the late-stage immune cells.
Oh, really? Where were they?
They found that both of these conditions share a genetic vulnerability in a very specific, very early maternal cell population, specifically a maternal endometrial epithelial population.
Okay, what kind of cells are those?
They are cells that line the uterus, and what's crucial is that they express highly specific stem cell markers, specifically markers called POU5F1 and LGR5.
Wait, POU5F1 and LGR5. If they express those stem cell markers in the lining of the uterus, that means these are foundational cells. These are the cells that are present from the very, very beginning of the pregnancy, possibly before the embryo even implants.
Yes. If we connect this to the bigger picture, it changes everything we thought we knew about these traumas. It strongly supports an emerging concept known as endometrium spectrum disorders.
Walk me through that. How does an early flaw translate to a late failure? How does a defect in a POU5F1 positive cell in the uterine lining at week 1 result in a premature birth at, say, week 30?
Think of it like pouring the foundation of a house. If the concrete is mixed poorly on day 1, the house might look perfectly fine while you frame the walls and put on the roof. It might look fine for months, but once the house is fully loaded with furniture and a big storm hits, the structural integrity of that bad foundation finally buckles.
Ah, I see.
Right. If the mother's foundational uterine cells are flawed before conception, the placenta they help build might function adequately when the fetus is tiny. But as the fetus grows and demands massive amounts of blood flow in the 3rd trimester, the poorly built interface simply collapses under the biological weight.
The foundation was cracked before the walls even went up. That is heavy. But at the same time, it is incredibly empowering. Because if we know exactly which maternal cells carry the risk and we know they are present from the very beginning, it opens the door to predicting, screening for, and eventually treating these conditions in a way we never could before. We don't have to wait for the house to collapse.
Exactly. It moves the entire field of obstetrics from reacting to late-stage symptoms to understanding the exact cellular mechanics of the failure before it ever happens.
So let's step back and look at the whole picture. If I'm synthesizing everything we've talked about today. We started by zooming in to a 0.5 micrometer view of a single fetal stem cell. We watched an epigenetic toggle switch flip, suffocating the cell's peaceful nature and turning it into an invader using HLA-G and FN1. We watched that cell march into maternal tissue to violently but necessarily rip up and pave over her arteries, displacing maternal cells through those R0 to R2 states.
A completely wild process.
And while all that's happening, the mother's DSC3 cells deploy endocannabinoid stop signs to keep the invasion in check. And finally, we saw how mapping these specific interactions reveals that diseases like pre-eclampsia and preterm birth aren't spontaneous mysteries. They are failures of this intricate cellular negotiation. So what does this all mean?
It's the ultimate question.
For me, this atlas shifts our entire perspective on human biology. We can no longer look at early development or pregnancy complications as independent biological events. We have to view them as a relationship at a shared border. And like any high-stakes relationship, if both sides aren't communicating perfectly, if the signals are dropped or the brakes fail, the entire system falls apart.
That is the perfect takeaway. And I want to leave you, the listener, with a final thought to mull over. Consider the fact that the maternal-fetal interface is essentially a temporary disposable organ. It is built by two entirely different individuals working together. It sustains a life, and then it is discarded. If a cell-by-cell map of this incredibly brief temporary organ can predict the origins of lifelong diseases and severe complications, what other transient cellular interactions in our bodies are quietly determining our long-term health?
Oh, wow.
Right. Think about the temporary cellular scaffolding that forms when you heal a deep cut, or the transient immune cell communities that gather to fight a winter cold and then disperse. What secrets are they holding? How many of our chronic illnesses are rooted in temporary cellular negotiations that failed years ago?
That is a fascinating question to end on. We'll start this deep dive talking about the comfort of a simple static medical diagnosis. But human biology is anything but static. It's a dynamic, moving foundation driven by constant microscopic negotiations. I encourage you to think about the incredible, violent, and perfectly balanced biological negotiations that built you. Thank you for joining us on this deep dive into the blueprint of human development. We'll see you next time.